In looking at horizontal transformation, we are going to start off with the donor cell. With the donor cell, we are going to pass off DNA from the donor cell to the recipient. With that, we are going to be able to create new organisms with new genetic traits within horizontal gene transformation. In the different processes, we are going to look at transformation, and with transformation, we have a donor cell that contains the DNA within the cell, and then we will have a recipient cell that also contains DNA. During transformation, there are some steps that we must follow. In the first step, the donor cell dies, going through lysis of the cell. So in this process, we become a naked DNA because our membrane is destroyed. In the naked DNA, the DNA is later released into the environment where it can potentially break into pieces within the environment. We then have the recipient cell that picks up, so it'll grab a piece, the naked DNA, incorporating it into its environment where the DNA can make this recipient into different things that can be done within its cell. But we need to know that not all of the cells can pick up this naked DNA. We have to have cells that are competent cells, and these cells are specialized proteins that are able to transport, again, the DNA across the cell envelope into the recipient. When we look at the different donor cells and recipients, they can be separated through this process for quite a bit of time and distance. We do need to know that the donor cell is dead, but the DNA is stable, allowing for the process of transformation to occur at any time for the recipient to get the DNA from the naked envelope into its cell where it can make the recipient cell do different things within their environment.